So, I'd like to thank the meeting organizers for inviting me to speak at this special symposium, which celebrates the life science research in honor of Dr. Rui Wu. And as Professor Zhang mentioned, I'm one of many life scientists who really have benefited tremendously from Dr. Rui Wu's effort in helping Chinese students. I came to the States in 1983 through Dr. Wu's CASPIA program and went to Cornell for my PhD study. At Cornell, I was fortunate to have had many interactions with Dr. Rui Wu. And this is a photo taken in the summer of 1985 at Dr. Rui Wu's student Xie Yong's wedding. Like Dr. Sun Xiaohong, Xie Yong, and other students at Cornell, I benefit tremendously from Dr. Rui Wu's mentoring. I not only learned from him how to become a good scientist, but also how to become a decent human being with integrity and humanity. And Dr. Rui Wu did this not by lecturing, but by his own behavior, treating everyone with respect and kindness. As you can see, probably that he's talking to me right here. So he was truly a role model in every way. His spirit lives on in all of us. And I'm delighted to see that all of us have been honoring Dr. Rui Wu's legacy by our own contributions to the society, as is exemplified by many former CASPIA students, and now by a new generation of young scientists represented by the recipients of the Rui Wu Prize. So I'd like to take this opportunity to congratulate this year's 10 recipients. Your exciting discoveries would make Dr. Wu very proud. So today I'd like to talk about a new gene regulation mechanism mediated by a class of RNA binding proteins called the pIRI proteins and their partner RNAs called the pIRI interacting RNAs, or piRNAs for short. I will focus on how the pIRI-piRNA pathway functionally link different constituents of the genome together via post-transcriptional regulation. So as you all know, our human genome contains 23,000 genes. In addition, our genome contains vast intergenic sequences in which about 1 million of transposons reside. And 90% of these transposons are retrotransposons, which are transcribed into messenger RNA, into RNAs in order to transpose. And transposons today are still viewed mostly as selfish parasites that invaded our genome and that we just have to live with. In the past decades, several labs work, including that of my lab, have shown that in addition, there are a large number of non-coding RNA coding genes now in the genome, including those genes encoding tens of thousands of link RNAs, as you've heard from this symposium, and also multi-millions of piRNAs. Finally, we should not forget that in our genome, there are also 14,000 pseudogenes. These pseudogenes mostly are transcribed. However, they cannot be translated due to the mutations in the coding region. Pseudogenes are viewed today as a defunct carcass of active genes on their way out during evolution. So despite the conspicuous presence of these major constituents of the genome, very little is known about their regulatory relationship. And today I hope to show you that transposons actually play a very active role regulating the expression of protein coding genes. In addition, I'll show you that pseudogenes also play an active role in regulating the expression of protein coding genes. Furthermore, I will show you that transposons can also regulate the expression of link RNAs. And finally, I hope to demonstrate to you all these regulations are mediated by the piRNA pathway, in the germline at least. And finally, we should not forget in our genome, there's a last territory, namely centromeres and telomeres. In recent years, my lab and others have shown that centromeres actually also produce RNAs. And today I hope to show you that the piRNA pathway can also regulate the centromere function by regulating the centromeric RNAs. So all this work started about 23 years ago when my lab discovered the Argonauts gene family. So this gene family encodes proteins that are highly conserved during evolution with 4 functional domains. The central PAZ stands for PV argonauxiliary PAZ domain, which is illustrated in 3D image here in pink, and the mid domain in golden color here. Together, they bind to small RNAs, whereas the PV domain in the C terminus, which is shown in green here, actually resembles RNA-SH, which will become very important later for my talk. So this gene family can be divided into argonaute and PV subfamilies. As you probably know all very well, the argonaute subfamily proteins play a central role in the SI and the microRNA pathway. And they bind to siRNAs and microRNAs, which are about 21 nucleotides long. And these RNAs and these argonaute proteins are expressed in most cell types. Therefore, RNAi can work in most types of cells as a housekeeping mechanism. And so are the microRNA mediated regulations. So in contrast, another subfamily protein called PV subfamily proteins, they are mostly expressed only in the germline and in the primitive stem cells. And they bind to yet another class of non-small coding RNAs that we discovered in 2006 and named PV interacting RNAs, or piRNAs for short. I must mention that around the same time, 3 other labs, namely Greg Hannon's lab, Tom Tuchel's lab, and Bob Kingston's lab at Harvard also discovered piRNAs within 6 months of our discovery. piRNAs are a bit longer than the microRNAs. And in addition, as I said, they are also mostly expressed in the germline and primitive stem cells. So this slide gives you a brief comparison of the salient features between piRNA and microRNAs. In addition to being a bit longer, piRNAs exist in great complexity. So today, my lab has cloned 16 million different species of piRNAs from mammalian system or even invertebrates, which is about 700 times more than the total known number of genes in our genome. And this is, again, is in huge contrast to microRNAs, which are only about 1,000 species. In addition, piRNAs correspond to all types of genomic sequences, in contrast to microRNAs, which mostly corresponds to the 3' UTR of messenger RNAs. And piRNAs are mostly made from long single-stranded precursors, in contrast to microRNA, which are made from hairpin structures. So this finding was very exciting to us because, as you know, the entire field of molecular biology has been focused its research on the central dogma in the past 6 decades. The discovery of microRNA and siRNAs further enriched this dogma by telling us that now there are 2 new mechanisms mediates the stability, control, and translational efficiency of messenger RNAs. However, we all know that the central dogma only covers about 1% of our genome. How about the vast majority of the genome that used to be called the junk DNAs? So we found actually most of the piRNA encoding genes are hiding in these intergenic regions. So this finding was really exciting to us because you likens our genome as the world, then the traditional genes are like the old world. And suddenly we found ourselves landed ashore of a completely new world with different kind of genes encoding different kind of RNAs. And this was a pie I baked for my lab to celebrate the discovery of piRNA. And this discovery was rated by Science as one of the 10 breakthroughs that year. So this hooplas aside, an important question remains. Are these piRNAs having any functions, or they're just the RNA equivalent of genetic junk? To study the piRNA function, we have been working on their nuclear function and cytoplasmic functions. Today, I will only focus on their cytoplasmic function. This work was done in mice using spermatogenesis as a model. As you probably all know that mouse spermatogenesis started by rapid division of germline stem cells that produce many spermatogonia. And these spermatogonia will then enter meiosis, become spermatocytes. After 2 rounds of meiotic divisions, the diploid 4C spermatocytes now will become haploid 1C spermatids. These wrong spermatids will then undergo drastic morphogenetic process to change themselves into mature motile sperm. So there are 3 PV genes in mice, and they are all expressed during spermatogenesis. The first one, called MIV2, is expressed in the stem cell region, and the second one, called MILI, is expressed from stem cells all the way to early meiotic cells. And the third one, called MIV, is only expressed in meiotic cells. Corresponding to these two different, the 3 different kinds of proteins expression, there are 2 different types of piRNAs expressed during spermatogenesis. The first abundant population is called the spermatogonial piRNA, which is expressed mostly in the stem cell region, and they mostly target transposons. That led to the famous function of the piRNA pathway in suppressing transposons jumping. However, when we discovered the targeting piRNAs, to our surprise, only less than 40% of these piRNAs actually target transposons. So the question becomes, what's the function of these non-transposon piRNAs? To address this question, we decided to use MIV mutant as an entry point, because in the MIV mutant, most of piRNAs are no longer produced. So we can study without piRNA, what kind of cells function will be deficient. And we know piRNAs collectively are probably very important, because in the MIV mutant, the meiosis will be blocked at the exit stage. So then we wanted to know where MIV are expressed. Our earlier work has shown that MIV is actually a cytoplasmic protein that's specifically expressed in the packaging stage spermatocytes. As you can see here, MIV is a red protein only in spermatocytes, but not in germline stem cells, which are in the basal layer of these 14 DPP testicular seminiferous tubules. In order to see if MIV regulate function of genes at the transcriptional level, since they're in cytoplasm, we isolated these MIV expressing cells and the equivalent cells from wild type and the MIV mutants. And we did the transcriptome profiling, and as you can see, to our surprise, in these mutant meiotic cells, namely spermatocytes, over 400 genes become significantly overexpressed as compared to their heterozygous sibling cells. So this analysis indicates that MIV somehow is important to suppress overexpression of these messenger RNAs. Now, given that MIV is important in piRNA pathway, we wondered if this is involving piRNA as well. To address the question, when we knock out another gene called the MOF10L1, that's also important for piRNA biogenesis. And we did the transcriptome analysis, and we found a very similar transcriptome shift in these MOF10L1 meiotic cells. So this analysis allowed us to conclude that the MIV-mediated piRNA pathway is very important to suppress the overexpression of these meiotic genes in primary spermatocytes. If that's the case, we would expect for those messenger RNAs which are targeted by the MIV piRNA complexes, they will contain the piRNA target sequences. And those are not the target, they may not contain the target sequence. So that indeed is the case. When we analyze the transcriptome of MIV plus and minus spermatocytes, we found that only those messenger RNAs which are upregulated in the MIV mutant, they contains large number of piRNA target sites, and the rest of the messenger RNAs actually contain very few target sites. So these data allowed us to conclude that MIV regulation of messenger RNA is guided by piRNAs. We then wondered what kind of piRNAs are there to serve as a target site, and turned out that those piRNA target sites themselves are transposon sequences. This was very surprising to us because we thought we understand the transcriptome and our genome so well, the entire field. However, no one before our finding actually reported that there are transposon sequences hiding in many messenger RNAs. And so we can conclude that the MIV regulation of messenger RNA is guided by transposon piRNAs. So then the question becomes, where are transposon sequences hiding in these messenger RNAs. When we analyze the transcriptome, I'm showing you here a meta map of a messenger RNA that representing the entire transcriptome with its 5' UTR here, protein coding sequence here, and 3' UTR here. As you can see, for those messenger RNAs, which are significantly upregulated in the MeWe mutant, they contain a significant number of transposon sequences. For those messenger RNAs which are upregulated, but not as significantly, they contain fewer transposon sequences. And for those messenger RNAs that are not upregulated in MeWe mutant, they do not contain transposon sequences. So then we wanted to know what kind of transposon sequences are there hiding in the 3' UTR or so many messenger RNAs. Turns out more than 50% of them are the sign elements, and there's also a fair share of LTR line elements and other repeats. So now the question becomes, does messenger RNA degradation actually occurs at the piRNA and MeWe target site, given that PV protein, some of them have slicing activity due to the presence of their PV domain? And to address the question, we focused on individual messenger RNAs. For example, this is a TDRD messenger RNA, which is a target for the piRNA MeWe degradation, with 5' UTR here, the protein coding sequence here, and the relatively long 3' UTR here. As you can see, indeed, there are two sign elements hiding in the 3' UTR. Now, if you zoom in into this sign element and do deep sequencing, which we did for this messenger RNA and the rest of the transcriptome, we found this cleavage site right at the binding site or predicted binding site of piRNA in this sign element. And this cleavage site is about 50% in depth because this MeWe cells we used is the heterozygous MeWe cell. There's only one copy of MeWe there. So this suggested to us that MeWe may degrade these messenger RNAs at its binding site in a dosage-sensitive fashion so that with one copy of MeWe, you can degrade 50% of the RNA molecules. And this is true for all three replicates. Now, if this is really true, we reason that if you can mutate the slicer residue that's important for MeWe slicing activity, then you should no longer see the cleavage. And that indeed is the case. When we just mutate the two amino acids, which are important for the slicer function of MeWe, we completely abolished the cleavage activity of MeWe towards this messenger RNA. Now, when we look through the entire genome to find out all those messenger RNAs which are cleaved by MeWe in the heterozygous meiotic cells and line them up with their target piRNAs, as you can see, piRNAs are somewhat heterogeneous in size, mostly ranging from 28 nucleotides to 32 nucleotides. However, when you line up the 5' UTR or 5' end of piRNA with its target messenger RNA, you see it's always at the 10 nucleotides away from the 5' of piRNA, the cleavage happened. So this allowed us to build a model, propose that the MeWe and piRNA complex will directly bind to target messenger RNA 3' UTR, and this binding leads to MeWe cleavage at the 10 nucleotides away from the 5' end of piRNA. Now, if this model is correct, one can imagine that if you build a reporter gene that expresses a transcript that contains a 5' UTR, a coding region encoding a reporter protein N-challenge, and a 3' UTR that insert a sign element that's flanked by LOXP sites. You can then also cross out this sign element in a transgenic strand where the Qui protein is expressed in the meiotic cells and to produce the same messenger RNA at the same abundance, except for that the sign element now is no longer there. Now you can ask the question, we're inserting sign element here, reduce the N-challenge mRNA's expression level in meiotic cells? And if so, is MeWe involved in the generation of this messenger RNA? So the data is shown here. As you can see, in these meiotic cells containing a sign element, and in these meiotic cells, this messenger RNA no longer contains a sign element, but otherwise they're expressed in the same level. And the expression level of this messenger RNA with sign elements is indicated in blue, and this one is in red. you can see that the pre-messenger RNA level of these two messenger RNA is identical or statistically identical, which suggests that inserting a sign element does not change the transcriptional rate of these messenger RNAs. However, when you remove the sign element, you see the mature messenger RNA become overexpressed significantly. So this analysis allows us to show that indeed overexpression is controlled by piRNA. How about MeV? Now, if you introduce this messenger RNA, still contains a sign element in the MeV mutant as compared in the wild type meiotic cells, even though sign element is still there, but because of lack of MeV, the messenger RNA again becomes significantly overexpressed. So this analysis allowed us to show that MeV is directly involved in regulating the stability to prevent the overexpression of this messenger RNA. Encouraged by this, we then deleted transposon elements in the 3' UTR of several endogenous genes, and without any exception, such deletion all causes overexpression of the messenger RNAs. So this analysis allowed us to discover a very new pathway in which transposons can, through piRNA pathway, regulate the expression of many protein coding genes at the post-transcriptional level. Now, when we looked through every gene carefully to see if their messenger RNA's overexpression is corresponding to their containing of transposon sequences, turn out there are some exceptions. Namely, some messenger RNAs, they are overexpressed in the MeV mutant. However, they do not contain transposon sequences. So the question becomes, then what piRNA sequences regulate their degradation? To address the question, again, we focused on individual genes. For example, this messenger RNA encoded by STEMP-BP gene is a messenger RNA with its 5' here, protein coding region here, and the 3' UTR here. So you can see that this messenger RNA indeed is targeted by several groups of piRNAs. However, this targeting is very different in two ways. First, the piRNAs are no longer only targeting the 3' UTR, but they're targeting throughout the entire length of the transcript. Second, those piRNAs, they do not look like any transposons. However, they look almost identical to the antisense strand of the STEMP-BP gene, with the exception of a few polymorphic nucleotides. So these data allowed us to reason that these piRNAs are actually not produced by transposons, but likely by a pseudogene of STEMP-BP gene somewhere hiding in the genome. So when we searched the genome for the pseudogene of STEMP-BP, indeed we found such a pseudogene. We call the STEMP-BP pseudosequence. Now the situation becomes very interesting. If these piRNAs are indeed produced by this pseudogene, then when you knock out this pseudogene, you expect this piRNA to be reduced to background level, and that indeed is the case. When we knock out the STEMP-BP pseudogene, the abundance of piRNA reduced to almost background level. Then you would further expect that then the expression of this sister or cognate active gene will become upregulated because RNA become over-stabilized. And that indeed is the case. When we knock down the pseudo gene of STEMP-BP, the pseudo STEMP-BP's real RNA now is increased by 12-fold. This increase, again, is due to increased stability, because when we looked into the pre-messenger RNA of pseudo STEMP-BP mRNA, there's no change in the expression. So this analysis allowed us to conclude that some pseudogenes, they can produce antisense transcript, which fits into the piRNA pathway to regulate the expression of the cognate active genes. Now, encouraged by these findings, we wondered whether transposons will also regulate the expression of other RNAs, such as link RNAs. So as you probably heard from several talks in this symposium, there are many link RNAs. They're most expressed in a tissue-specific fashion. Several years ago, my lab profiled the link RNAs from all major tissues in mice. As you can see, the expression almost becomes a signature of the tissue. Let's focus on the testis here. Testis contains over 15,000 link RNAs, And meiotic cells alone contain over 6,000 linked RNAs. If we do a more careful profiling of testes according to the age of the male mice, so that you can find out different stages of cells at different time, and you can see that meiotic cells, namely spermatocytes, contain over 6,000 unique linked RNAs. We wondered whether the expression of this linked RNA will be also controlled by MeWe. And when we did the profiling analysis, indeed, we found out more than 1,000 linked RNAs become significantly overexpressed in MeWe mutant meiotic cells versus MeWe heterozygous meiotic cells. When we looked into these linked RNAs, which are overexpressed, indeed, they all contain transposon sequences. Excuse me. So this analysis allowed us to conclude that just like the situation of messenger RNAs, these linked RNAs, they are subject to the regulation of transposons through the piRNA pathway. So up to now, to summarize what I told you so far, let's review the central dogma, which has been used by all of us as the mainstream regulation of gene expression, namely a gene transcribed into a pre-messenger RNA, then got capped, tailed, and spliced and become mature messenger RNA and exported into cytoplasm. So this sounds right. However, it's really very partial a picture in terms of gene regulation. Our studies suggest that we should be cognizant of the fact that in germ cells and possibly other primitive stem cells, the transposons are also expressed. And these transposon RNAs will be processed into piRNAs together with MeWe protein and possibly other PV proteins. They'll bind to those messenger RNAs that contain transposon sequences in the 3' UTR to regulate their expression. We should also be cognizant of the fact that the pseudogenes are mostly active, and some pseudogenes can make antisense RNAs that will be processed into piRNAs, and together with PV proteins, they will target their sister active genes to regulate their expression. Likewise, for the expression of linked RNAs, it's not only illustrated here, but they are also subject to the regulation of the transposon originated the piRNA pathway. So up to now, I basically covered all the major constituents of the genome, except one final frontier of the genome, which is the centromere or telomere. Several years ago, we started to study the function of centromere and possible regulation by PV proteins. And because we found that actually centromeres are actually transcribed into RNA too. And centromeres, as you know, is very important for cell division. And in meiotic cells, they are very important for meiosis. A key regulator of chromatin segregation in meiosis is really the spindle assembly checkpoint. That's a cell surveillance pathway that avoids precocious anaphase onset until all the kinetochores are aligned correctly together to the spindle microtubules. Then if you have a prolonged spindle damage, then you cause aneuploidy, and these cells eventually will undergo cell death, as you can indicate from this tunnel staining of these meiotic cells here. So we actually found out MeWe mutant indeed has a significant increased meiotic cell death, which led us to think maybe MeWe is involved in regulating the centromere or kinetical structure. To probe that possibility, we looked into meiosis in wild type and in MeWe mutant, using the anti-phosphor histone 3 antibody to line up these meiotic chromosomes and anti-tubulin antibody standing right here to highlight meiotic spindle. As you can see, wild type of meiotic cells have these beautifully aligned spindle metaphase plates. However, in MeWe mutants, very often we see chromosomes are lagging behind. And when we quantify that, about 1/3 of meiotic cells actually show this big problem. So we then wanted to know, then what's the function of MeWe related to this meiotic division? And so let's briefly review the centromere and paracentromere structure in chromosome. So centromere and paracentromere, actually, they are composed of minor satellite repeats and major satellite repeats correspondingly. So the major satellite repeats that's made of paracentromere, these are highly repetitive short copies of repeats. There are about 200,000 repeats, which constitutes about 3% of the genome. And the minor satellite repeats is made of these 120 base pair repeats in many, many copies, about 50,000 copies. They made up of centromere and they composed about almost 0.5% of the genome. And both minor and major repeats can produce repeat RNAs, we call satellite RNAs, both in antisense and sense directions. So there are 4 populations of satellite RNAs. The antisense minor satellite RNA and sense minor satellite RNA from the centromere, and the sense major RNA and sense antisense major RNA from the paracentromeric region. And these RNAs are very important for the formation of paracentromere structure and centromere structure, which contains special proteins like SAMP-C. And in the paracentromeric region, they recruit histone methyltransferase SUVA39 to methylate H3K9 to form heterochromatin here. Now, this formed heterochromatin in the paracentromeric region will further be bound by cohesins to form a unique paracentromere landscape. And the centromere DNA will be bound by SAMP-E and then other kinetical proteins. So we then wondered whether MeWe will be actually regulating the metabolism of these satellite RNAs. Just to give you a bit background information, these satellite RNAs, they are not transcribed from any definitive promoter. Therefore, they are very heterogeneous in size. And also, they are mainly expressed during the DNA replication stage and accumulate till the meiotic division phase. So we then did the northern analysis to see whether these satellite RNAs are indeed become overexpressed in the MeWe mutant. And that indeed is the case. As you can see, this is the MeWe heterozygous meiotic cells probe for satellite RNA. They're expressed at the obvious level, but in the MeWe mutant, the expression level is significantly increased. And we wondered if this increase is really due to, in the MeWe mutant, there's no slicer activity. If so, if we just create a slicer deficient MeWe mutant mice in those meiotic cells, then the satellite RNA should be also equally overexpressed. And that indeed is the case. So in this way, we showed that the sense major satellite RNA, their overexpression is suppressed by MeWe. And turned out that's also the case for the sense minor satellite RNA and for the antisense major and minor satellite RNAs, as you can clearly see here. So this analysis allowed us to conclude that MeWe slices all satellite transcript to prevent their overexpression. So now the question becomes, what's the effect of overexpressing satellite transcripts on chromosome segregation? We first looked into whether overexpressed satellite RNA will be localized in wrong places in the chromosome, and that turned out is not the case. We also looked into whether overexpressed satellite transcripts will not affect kinetical structure or not. It turned out the kinetical structure is still normal. Then what's the problem? Then the problem we thought about is it could be this overexpressed satellite transcript will affect kinetical pairing, which is needed for meiosis. So kinetical pairing refers to the two sister or homologous chromosomes. Their connector will pair together, and so that during meiosis, they'll separate to the opposite end. And the kinetical is made of two structures, the outer plates, which is marked by this BERP1 proteins and many other proteins, and the inner plate that can be marked by CREST protein and many other proteins. The CREST protein representing inner plate, the inner plate will attach to the centromeric DNA, whereas the outer plate will attach to microtubules, therefore kinetical function as a segregation apparatus. So kinetical structure can be visualized in this way when they are paired between the homologous chromosome both the outer and the inner plates, as you can see, they become overlapped, become one spot. So we call that overlapped outer plate and overlapped inner plate configuration. And they are flanked by the major satellite RNA, which are the paracentric heterochromatin. And you merge them, you see beautiful canonical flanked or sandwiched by paracentric heterochromatin. Now, you can also expect there are other configurations. These abnormal configurations, such as a separated canonical outer and separated canonical inner plates, that will obviously screw up the chromosome segregation. Or you can imagine there are split outer plates but overlapping inner plates, or vice versa. They will also affect chromosome segregation. And now let's see in the wild type and MeWe mutant meiotic cells, how often we can see these kind of normal and abnormal things. So it turned out in the wild type situation, most of the time, the canonical paired very nicely. But in the MeWe null mutant or the slicer deficient mutant, you see a significant decrease in the canonical pairing. And yet there's a significant increase in this abnormal pairing. So this gives us a hint that the canonical pairing requires MeWe slicer activity, because MeWe slicer activity is important for both pairing the inner and outer canonical. The second conclusion we can make is that the overexpression of satellite RNA due to the deficient of MeWe will actually cause abnormal homologous canonical pairing, which will then eventually lead to aneuploidy, because that overexpressed satellite transcript will mess up the pairing, leads to abnormal chromosome segregation. So to further answer the question, how can this pairing will affect the canonical segregation? We thought that the MeWe pairing have a significant effect, but still not up to 30%. So we wonder whether the pairing also involved the dicer, because some of the antisense RNA from the satellite region, we envision they can be double-stranded, and that will be subject to dicer degradation. And turn out that's the case. As you can see, for this sense major satellite RNA, when it's in the wild type meiotic cells, they're expressed at low level. In the MeWe mutant cell, they become overexpressed. And in dicer mutant, they also become overexpressed. Now in MeWe and dicer double mutant, they can become significantly overexpressed. And that's also the case for 3 other types of RNAs. As you can see, they all become most overexpressed in the MeWe and dicer double mutant. So correspondingly, in these MeWe and dicer mutants, we see even higher number of meiotic cells have chromosome segregation defects. So as you can see here, the normal pairing canonical pairs in the wild type is near 100%, but in MeWe deficient and dicer deficient meiotic cells, they can become correspondingly decreased. And in double mutant cells, they become very low. Yet the abnormal pairing configurations become significantly increased in the double mutant. So this analysis allows us to conclude that MeWe and dicer have additive effect to the pairing of canonical during meiosis. So this analysis, and I don't have time to show you many other data to support this, but allow us to propose this model. We think that the satellite RNAs transcribed from the centromeric and paracentromeric regions are very important for forming normal paracentromeric and centromeric structure, but they cannot be overexpressed. Normally, in the presence of MeWe and dicer, they are expressed at appropriate level and just sufficient for chromosome to take up, but no abundant excess. Now, in either MeWe or dicer mutant, the excess RNA will be causing trouble. Therefore, in the normal wild type situation, this excess RNA, when they are in double-stranded form, they'll be cleaved by dicer. And when they're in single-stranded form, they'll be cleaved by MeWe piRNA pathway, and we've identified specific piRNAs that are against these satellite RNAs. And these cleavage products will form short single RNAs. These single-stranded RNA will sometimes adhere to the single-stranded precursor here to allow further degradation by dicer. And this eventually leads to the prevention of overexpression piRNA, which guarantees the accurate chromosome segregation. So to summarize my talk, again, I like to use this analogy. If you liken our genome as the world, then the traditional genes are in the old world. However, we should not forget that in old world, we also have pseudogenes and have transposons. So now the studies from early on my lab, and now many labs show that there's a big new world that contains many other new kind of genes that are coding for piRNAs, linker RNAs, and other non-coding RNAs. Today, I hope to have shown you that these two worlds are actually tightly connected. I've shown you that even the last territory of our genome, centromere, is also connected. Specifically, I've shown you that piRNAs can regulate the expression of traditional genes. piRNAs can regulate the expression of many linker RNAs, and the transposons can regulate the expression of many traditional genes, and even pseudogenes can regulate the expression of many sister active genes. And finally, piRNAs can also regulate even the function of the centromeres. So I'd like to start by thanking Toshi Watanabe, a former postdoc in my lab, and Jialing Xie, a current postdoc in my lab. Most of the results presented in this talk are completed by these two excellent postdoctoral fellows. And I'd like to stop here and thank you very much for your attention.
Thanks, Dr. Lin, for the wonderful talk. Any questions?
Hello.
Hi.
Hello? Hi.
Yes.
May I ask a question?
Yes, please.
Hello. Hi, it's Guoliang Xu.
Hey, Guoliang, how are you? I cannot see you.
I enjoy your talk.
Thank you for coming to my talk.
Yeah, I'm in New York, so it's the same time.
Oh, you're in New York. Okay.
Okay.
Well, come to see me again.
My question is that have you done conditional knockout of the MIV gene in the haploid stage? Is there any specific effect there?
Yeah, that's a very good question. We have not done that because so far we have not found a haploid specific CRE line yet. But once we have that kind of line, we'll be interested in doing that. That's a very good question.
Okay, thank you.
Hi, Fan. This is Hong.
Hi, Hong, how are you? Are you in Beijing or in Shanghai?
No, I'm in Shanghai.
Okay,
So,
great to hear from you.
yeah, this is symposium. So this is second time I listened to your talk in the
in a few months, yeah.
6 months. So I'm wondering, especially the last portion of your talk, how piRNA control centromere function, which is critical. You show most of the phenotype, if I understand correctly, is in meiosis process, right?
Yes, because we only knock out the piRNAs in meiotic cells.
Yeah, how about mitosis?
Yeah, I'll be very interested in doing that because in stem cell, we are now started moving early on to the germline stem cell region to see if there's a function in mitosis. We don't know yet, but your question again is very good. Yeah.
And so if it also control somatic cell mitosis, then that could has a major impact on chromatin instability, especially cancer.
Yes. Yeah, precisely. So that's very interesting. In normal somatic cells, in most of the differentiated cells, because the PV proteins are not expressed, piRNAs are not expressed. So we believe there's a different mechanism. Possibly dicer become more important. However, in cancer cells, as you said, actually many type of cancer cells shows overexpression of PV proteins. And the transposons are also become active in some cancer cells. And so one can imagine piRNA might be produced, and there are some reports saying piRNAs become produced in cancer cells. Mm-hmm. So in that case, will that actually become a negative effect to make cancer cells have increased the frequency of aneuploidy by messing up the cell division system? It's quite possible.
Yeah, especially recently, there's realization some cancer has so-called chromatin catastrophe. Basically,
Yes.
there's many fragmentation and rearrangement, and the mechanism so far is not that clear.
Right, right. Yeah, I think that's a very interesting new area of cancer research. And we have not done much, but I've been also following the literature a bit. It can really explain why cancer cells are so heterogeneous.
Yeah.
And different populations, it's a cell selection process, right? You start with some pioneering cells, but they may not last long. And some other cancer cells with some other karyotype will take over. Yeah.
Thank you.
I wish I could be with you guys in Shanghai, but the pandemic has been a big toll here.
Hi, Fan. Great talk.
Hi.
So I have a interesting feeling that the PV and piRNA system is kind of like the CRISPR-Cas, but it's not CRISPR-Cas9, but like Cas13 because it knocks down RNA.
Yes.
So you think,
yeah.
Like a mammalian version of immune system that to protect the germline? Can you comment on that?
Yeah. It's yes. I think that's a very good analogy. It's more like the immune system at the genetic level, yeah, or the RNA level. I do agree. Actually, some immunology friends of mine at Yale, they suggested similar idea too, and they even thought they might be involved, possible even in somatic cells in like a VDJ recombination. But I actually Rosalind was trying to collaborate with me to work on that, but we haven't really pushed the collaboration very far yet.
Yeah, and also
can
you comment on why so many piRNA exist in the germline, but not?
Yeah, it's crazy. That's a good question. I think because there are so many functions, and most importantly is the epigenetic function. I didn't have chance to talk today. I only talked about the cytoplasmic function, that's post-transcriptional regulation. And one can imagine for transcriptional regulation, for post-transcriptional regulation, you really don't need multi-millions of piRNAs, right? Because you're targeting the transcriptome, even including transcriptome for other non-coding RNAs and linker RNAs, is still much smaller than genome. However, when the piRNA population and PV proteins are in the nucleus, they actually become a guidance mechanism to guide epigenetic factors to different sites. And epigenetic programming happens throughout the entire genome. And when you calculate that, they really need the piRNA and many, many piRNAs. And also piRNA, the length is perfect for that because our genome is about 100 times more complex in transcriptome. So if you have a siRNA or microRNA with 21 nucleotides, for a mammalian genome like human genome, 3 billion base pairs is too short to have any true specificity. But piRNAs are perfect. We actually have shown that if you, which we did both in Drosophila and mice, take out a particular piRNA target sequence and insert it into a ectopic region, and it will actually recruit, in the case of fly, the HP1 to that site. And in the case of, in mice germline, you can recruit DNA methylases to that site to cause methylation. And that's why you need a lot of piRNAs.
Okay, thank you so much.
I hope that makes sense to you.
Yeah.